Hey everyone, and welcome. Today, we are diving headfirst into one of the coolest parts of genetic engineering. We're going to look at the incredible toolkit scientists have built for one super important job: how to get a piece of custom-made DNA into a living, breathing cell.
So, I want you to imagine you're a programmer, but you're not coding a computer, you're writing code for a living thing. The biggest question you've got to answer is this: how do you actually upload your new program, your new DNA, into the unbelievably complex machinery of a living cell?
Okay, before we get into the how, let's just make sure we're on the same page with our key players. First, you've got the recombinant DNA. This is your precious cargo, right? The brand new set of instructions you've designed. And then you've got the host cell. That's your destination, the living factory you're trying to upgrade with your new software.
So how do we get past the cell's natural security system? Well, our first couple of methods are really clever ways of basically talking the cell into opening its doors, using a little bit of chemistry and physics.
First up is a method called transformation. Now, this is the absolute workhorse for bacterial cells. What scientists do is they give the bacteria a little bath in calcium chloride. This clever trick helps get rid of the static cling, the negative charges that make the DNA and the cell wall push each other away. Then, bam, a quick blast of heat, they call it a heat shock, and the cell's membrane gets a little leaky, just long enough for the DNA to sneak inside. It's a classic, it's cheap, but it's not super efficient. A lot of the cells just won't take up the new code.
All right, but what if you need a method that works on more than just bacteria? That's where electroporation comes in. It's exactly what it sounds like. You basically zap the host cell with a short, powerful electric pulse, and this jolt creates these tiny, temporary holes in the cell's membrane. The recombinant DNA can then just zip right through those openings before they seal back up. The really cool thing here is its versatility. This works on pretty much any kind of cell, from bacteria all the way up to human cells. The only downside? That electric shock can be a little harsh, and sometimes you end up damaging or even killing some of the cells you're trying to modify.
Okay, so sometimes just persuading the cell isn't going to cut it. You need a more direct approach. These next two methods are all about physical delivery, making absolutely sure that precious cargo gets exactly where it's supposed to go.
And this is microinjection. I want you to think of this as a super precise surgical strike at the microscopic level. You use an impossibly tiny glass needle to inject the DNA right into the nucleus of a single animal cell. You're bypassing all the defenses and going straight to the cell's command center. The precision is just unreal. But, as you can imagine, it takes a ton of work, it's slow, it's expensive, so it's not really practical if you need to change millions of cells at once.
And now for my personal favorite, because it sounds like it's straight out of a sci-fi movie: biolistics, or as it's better known, the gene gun. Plant cells have these really tough outer walls, right? So scientists came up with this idea. They coat tiny little bullets made of gold or tungsten with DNA, and then they literally shoot them into the plant tissue. It's a total brute force method, but hey, it works. The only thing is, it's a bit of a scattergun approach, and you can cause a fair bit of damage to the cells you're aiming for.
So we've seen some very hands-on methods so far. But what if we could just hire a natural expert to do the job for us? This last method is amazing because it uses a bacterium that has already been genetically engineering plants for millions of years.
I want you to meet Agrobacterium. This little guy is a natural wonder. It infects plants by cutting out a piece of its own DNA and pasting it right into the plant's genome. So scientists looked at this and had a moment of pure genius. They basically hijacked this process. They take out the bacterium's DNA, slot in their own custom-built recombinant DNA, and then just let Agrobacterium do its thing. It becomes this perfect, highly efficient delivery truck, or vector, carrying the genes we want right into the plant cell. It's elegant, it's effective, though it is pretty much limited to the types of plants this bug can naturally infect.
Okay, so we've just run through 5 totally different ways to do this, and each one has its own pros and cons. So how on earth does a scientist choose? Well, it really all comes down to the specific job. What kind of cell are you working with, and what exactly are you trying to do?
So this table is basically your cheat sheet for the entire toolkit. If you're working with bacteria, transformation is your classic, go-to, low-cost option. If you need a one-size-fits-all approach for any cell type, you're looking at electroporation, as long as you're okay with losing a few cells along the way. Need that guaranteed pinpoint accuracy in an animal cell? You've got to use microinjection. And for plants, you've got 2 great choices: the brute force of the gene gun or the slick natural delivery system of Agrobacterium. The main takeaway here is that there's no single best method. It's all about picking the right tool for the right cell.
So why does any of this really matter? Because these techniques, these are the absolute foundation of all of modern biotechnology. This is how we get bacteria to pump out insulin for diabetics. It's how we design crops that can survive a drought. It's how we develop gene therapies to fix devastating inherited diseases. We are literally, right now, learning how to rewrite the source code of life itself.
And that leaves us with a pretty massive question, doesn't it? We have these unbelievably powerful tools that let us edit the very fabric of living organisms. So as we go forward, the question isn't just can we do something, but what are the most important, the most ethical, the most world-changing challenges that we should point this amazing technology at next?